So here we are with our handy dandy pipe cleaner chromosome set and our cell membrane material, just to show us where the cell membranes are forming around our new cells in our meiosis process. So we're going to look at the process of meiosis, and as we go along, you're going to replicate it in your set. So meiosis has a lot of steps that are similar to mitosis, but you have to watch carefully because some of them are really different. And they're going to allow the differences that make our sex cells, like sperm and eggs, special. So let's start with our diploid cell. Here's our diploid cell where we start meiosis. All of our chromosomes are in the form of chromatin. They're just hanging out together. You can't see them under a microscope yet. But in meiosis, the first step is going to be prophase 1. And in prophase 1, what we're going to see is chromosomes are going to appear. And the homologous chromosomes, these chromosomes that have the same type of genetic information, but different flavors of those genes, or alleles we call them, are going to pair up. So here they are, they've paired up. This is prophase 1. Now in metaphase 1, you'll remember from mitosis that our chromosomes line up in the middle, but they're not going to line up in the middle like this, the way they did in metaphase 1 of mitosis. In meiosis, metaphase 1, they're going to line up in their homologous chromosome pairs right next to each other along the middle of my cell. Notice here that I could have put these two homologous chromosomes in this order, or these two in this order. And that's going to happen randomly in this process. And you can imagine if we had 23 pairs of chromosomes, the way a human cell would, there are lots of different combinations of how these different pairs of homologous chromosomes would match up with each other, or on which side they would end up. So here we are at metaphase 1. Now it's time for anaphase 1. In anaphase 1 of mitosis, we know that each of the sister chromatids would get pulled apart. But anaphase 1 of meiosis is different. Now each of the chromosomes of each pair are going to get split up. Here we are. So this is the end of anaphase 1. Notice the different parts of each pair have ended up on each side of the cell. Let's do telophase 1. Here's telophase 1. We create 2 new nuclei, and in cytokinesis, now 2 new cells. So notice that we started with 4 chromosomes in our diploid cell, and now we have 2 chromosomes in each of our cells that we've created in meiosis 1. So our chromosome number has decreased by half. Okay, now it's time for meiosis 2. Meiosis 2 looks a lot more like mitosis. So in meiosis 2, prophase looks very similar to prophase of mitosis. Chromosomes appear, and then in metaphase 2, they're going to line up in the middle of each cell. So my chromosomes have lined up in the middle. Now in anaphase 2, those chromosomes are going to be pulled apart, and this again looks very similar now to mitosis. So our chromatids, our sister chromatids, are now being pulled apart in anaphase 2. Like so. Now in telophase 2, and cytokinesis. Our 2 nuclei form for each of these cells. And so you're going to end up with 4 different cells. All of whom have 2 unduplicated chromosomes. So let's count them. 1, 2, 3, 4 cells, and each of those 4 cells have 2 chromosomes. We started with 4 chromosomes in our diploid cell, so that makes all of these haploid cells. And notice here that the genetic material in these different cells is different. So this has different genetic material from this cell.